Now we're going to continue our discussion of macromolecules, starting with giving the list of the different kinds: carbohydrates, lipids, proteins, and nucleic acids. We'll deal with these in the next chapter. We'll talk about these guys right now. Before we get to all the details that are in this table, which you'll draw in class, so we'll talk about it again, I want to talk first about what it means to put these guys together. So we have monomers, which are basically the building blocks of these different molecules. And let's see, zooming in, I don't know if that's a good idea. We're learning here. If you look at, there we go, a condensation reaction. That's a reaction when you're putting two monomers together. So we're going to be catalyzing this with an enzyme. An enzyme is basically a protein that helps to have reactions take place in physiological conditions in an organism. It's condensation because the enzyme is going to remove a OH from one side of the monomer, remove an H from the other side, and put it together to make a water when they make the covalent bond, hooking these two black-brown blobs together. This is a type of reaction that happens when we put amino acids together for a protein, and very specifically for this first section, it's going to be when we put two sugar units together to make a polymer or a longer sugar. So you can hook a string of glucoses together, so maybe this is glucose here, hook it to the next glucose, lose a water molecule, and make a covalent bond holding it together. When you do that multiple times, you now have what's called a polymer. Similarly, you can, I go this way, you can rip a molecule apart by a hydrolysis reaction. The hydrolysis reaction takes a polymer, again enzyme catalyzed probably. You can have chemicals that do it too, but most of the time we're dealing with living systems that use enzymes. Enzyme's going to break that bond, bring a water molecule in, break the water molecule apart, put an OH on one side and a hydrogen on the other side, and you continue doing that and you break it apart. This is similar to what goes on in your stomach or probably your small intestine in digestion, when you break apart polymers of foodstuffs and break it into the building blocks that you go and recycle in your body. I'm going to talk about starch and talk about the glucoses that get liberated in that. So let's go back down to full size. Sorry about this. I need to get my act together. There we go. Look at the next couple of slides. So, bottom line is hydrolysis is breaking apart. Condensation reactions are putting together. So, this happens to be an amino acid. I'm going to skip over it right now because we're going to talk about sugars. So the first macromolecule group we want to talk about are sugars. They are all ending with an OSE. They're oses. So when you have a 5-carbon sugar, the carbons are these big black balls. You have a pentose. The two famous ones that we're going to deal with this time are ribose and deoxyribose, part of RNA, part of DNA. And then a 6-carbon sugar is called a hexose. Guy that you know and love soon, if you don't already, will be glucose, 6-carbon sugar. Fructose is a 6-carbon sugar, although a stable conformation of it is this pentagon, and we're going to get into those differences later. Mannose and galactose are two other types of monomers. If you hook a galactose and a glucose together, you make lactose. And if you hook a glucose and a fructose together, you make sucrose. So getting up close to a little bit more to the structures, getting used to the organic chemistry structures, you start numbering your carbons with number 1. So here's carbon number 1, 2, 3, 4, and 5. He has 5 carbons. I'm going to show you a slide later on to show you why they make this ring. It's more stable thermodynamically. Deoxyribose is just like ribose, but they highlight here the two changes. It's missing an oxygen, hence its name deoxyribose. We're going to see those up a lot in our DNA molecules. Then getting in a little bit closer on our hexoses. Again, we're going to talk about glucose a lot. Here's carbon number 1, 2, 3, 4, and 5, 5 and 6. The sixth one is not part of that ring because it's just not thermodynamically stable, and instead one of the oxygens hanging off that carbon becomes part of the ring. Again, if you hook a glucose together with a fructose, you're going to drop the OH from this glucose and you're going to take the H from that fructose. You're going to make a bond between them, drop the water. It goes on an angle just because of these shapes. I'm going to show you that in class, how their shapes are made this way. You make a bond between them, you make sucrose. Sucrose is table sugar. That's the stuff that you bake with, you put in your iced tea, you find all over the place. So it's a combination of a glucose and a fructose. Fructose all alone is actually much sweeter than glucose. So why do they make that ring? This is sort of like higher level organic chemistry that you should be aware of, but you don't have to memorize what's going on here because I know it's a little intimidating. Basically, we normally see glucose in this linear form. Here's 6 carbons. It's an alcohol, right? Because it has the hydroxyl groups hanging off it, those OHs. This group up here is that carbonyl group, and it's at the top, so that's an aldehyde. So what's going to happen, and they highlight it very nicely in here, the OH that's hanging off the 5th carbon that's highlighted here is going to change bonds with that carbonyl. And watch what happens. See, there's a double bond of oxygen right here. It's going to grab that hydrogen that used to be on the alcohol group or the hydroxyl group here. And that hydrogen is going to jump over here and take the other bond from oxygen to satisfy the 2 bonds it needs, and then make a bond between the oxygen and that carbon and that oxygen and that carbon, making it a ring. It turns out that it is most stable to exist in a ring, so it does. Now, an interesting little side note here, when it makes this ring, and usually we drop the carbons and just put angles in there, sometimes that OH will be facing down, and sometimes it'll be facing up. It's a 50-50 chance. One is called the alpha form, and the other one is called the beta form. We're going to get back to them later on. Oh, I'm going to get back to it right now. So when the oxygen and the OH is down, it's an alpha glucose. It's a beta glucose if that OH ends up hanging up. It's going to be 2 different kinds of digestible or non-digestible sugars. So starch is a linkage, a huge polymer, thousands long. We're just seeing a snapshot here of glucoses linked together in the same way. So that oxygen's always down, and it's always the alpha glucose. So the bond is forced in this direction. Cellulose, on the other hand, starch is stuff that you find in your potatoes and your wheat. So it's in your pasta and all that kind of stuff. Cellulose, on the other hand, is what plants use to keep them stiff. It's in their cell walls. It is a combination of a 1,4 linkage of beta glucoses, so the OHs that are in the other direction, and it ends up making the sort of up and down thing. Cellulose is not digestible by humans. So if you eat grass, or if you eat, say, corn, you can't digest it. You can't digest large amounts of cellulose. It just passes right through you. This is technically fiber. But there are organisms who know how to digest, and that would be our friends the cows. And the cows have multiple stomachs. They move their food between the different stomachs, and they have resident bacteria inside them to help them digest that cellulose, and they can use it for food. So then we eat the cows who have already processed the cellulose, and everybody's happy. Just looking again at the cellulose, it makes these long linear chains because of the way the bonds are formed, and it makes fibrils. Starch, on the other hand, is branched. And then we also make in our bodies a similar but slightly different form of a polymer of glucose called glycogen. We store it in our livers, and we release glucose molecules when we need energy. There's lots of places that we have deposited starch. Again, this is the cellulose. You can see how fibrous it is. That's the cellulose, excuse me. The starch is in granules that are in potato cells, are showing you here. And then this is the glycogen in the liver cells of a monkey. So when you have a polymer of a sugar, you have a polysaccharide. Poly meaning many, and saccharide means for sugar. A monosaccharide would be the glucose that makes it up. I think I'll stop here with the carbohydrates, and the next video will be about lipids.